everybody. Thanks for joining us today. I hope you are fans of computer science, because today we're hearing from Daphne Koller, CEO and founder of In Vitro, machine learning drug discovery and development company. As I was telling Daphne, she's one of the most accomplished people we've ever had on these webinars, and it's hard to know where to start, but so I'll just sort of start at some point and go outwards. She was on the Stanford faculty for 18 years as a Rajiv Madhwani Professor of Computer Science, Chief Computing Officer at Calico Labs, and we were talking earlier about her interest in online learning. She co-founded Engagely and Coursera, which she led as co-CEO with Andrew Ng. She's author of over 300 publications with an H index of 146. It's pretty astonishing. I mean, it just goes on. So we'll get to that. I just have to mention a few more. Recognized as one of Time Magazine's 100 most influential people, inducted into the National Academy of Engineering, and received a MacArthur Fellowship, along with many, many other awards. I could be here for a while. Today, we're going to learn about machine learning for identifying translatable biomarkers and targets. Hi, Daphne.
Hi, thank you so much for the generous introduction, Casper, and thank you for inviting me to speak. So I'll tell you a little bit about our work for how we think about doing drug discovery differently using machine learning. So the fundamental premise behind our work is that many complex diseases today remain opaque because we simply do not define them properly. We define them using coarse, subjective clinical symptomology that sometimes dates back 100 years. The symptoms are often subjective, filtered through the lens of a clinician and a patient. And if we really want to uncover new medicines, we need to understand the diseases that we're treating. So we need to unravel disease biology and identify actionable causal nodes. The problem is, I can tell you, after a career of having worked in many applications of machine learning to a variety of domains, human biology is really complicated. It's the most complicated thing I've ever done. Far too complicated for people to ever fully understand in terms of our minds. And so the way that we have taken to address that is to generate large amounts of data that allow us to interrogate biology from diverse perspectives, and then use machine learning to interpret those massive amounts of data, which can help us redefine disease and hopefully identify effective therapeutic interventions. So to advance towards that goal, we built In Vitro, which is a new kind of drug discovery company. You can tell by the name, which is the integration of in silico in the computer, and in vitro, which means in the lab, really trying to create a company that is truly cross-functional across disciplines, so that together in collaboration, we build powerful infrastructure for generating and aggregating large amounts of data about human biology at different scales. We develop novel methods for interrogating those data, and we use those methods to make better predictions about the effects of interventions in a human organism, which ultimately will give us, we believe, or have already shown, a pipeline of insights and therapeutics, which help us address unmet medical needs in our core therapeutic areas, which are currently neuroscience, metabolic disease, and oncology. So the kinds of data that we bring together come in two different forms. There's cellular data that we generate in our own lab. Those come from human-derived cellular disease models that are biologically relevant systems. We measure those in a variety of different ways, some off the shelf, some quite proprietary. Those include various types of omics, fixed and live cell imaging, and a range of functional readouts. On the other side, we aggregate different types of clinical data from patients. Again, high content, unbiased measurements of human physiology, which includes various omics on blood and tissue, as well as multiple forms of imaging, specifically histopathology, as well as anatomical imaging. We take those different high content data that are far too large and complex for the human mind to fully understand, and we apply cutting edge machine learning techniques to basically learn the language of these models. So if you think about all of the current frenzy around large language models. This is a smallish language model for languages of specific types of biological modality, such as histopathology, cellular imaging, and so on, but it's still a language model. And then we take and use that language model to reinterpret the phenotypic consequences of human disease from the fuzzy definitions of disease as we use it today to something that is much more crisp and actionable. Now, in understanding disease is only the first step. Then we need to understand what happens to interventions in those biological states. And for that, we use genetics, because that is one of the few cases where we actually have ground truth for causality in human biology. Causality is really hard to come by, especially in biological systems. For us, genetics comes in two forms. There is genetic interventions that we make in the cellular systems in our lab using technology such as CRISPR. And on the other side, we have the kind of genetics that are experiments of nature, such as all of you and myself, which allow us to understand the causal connection between genotype and phenotype. So with putting those all together, we hope to discover targets which are high confidence genetic drivers that meaningfully modulate clinical outcomes in patients. Identify the right group of patients using a data-driven machine-learned biomarker approach that gives us coherent patient segments rather than trying to cure what is a heterogeneous set of causes that somehow manifest in a vaguely similar way, which is what we try to do today. Really identify the right coherent patient segments and hence inform our clinical strategy. And finally, of course, therapeutic interventions are molecules that modulate those targets in those patients to modulate the relevant outcome. And that, in turn, is what we use to build our pipeline of therapeutic programs. So the capabilities that we've built might seem disparate, but they really help us interrogate diseases across the spectrum of genetic architectures. We think of genetic architectures as lying on this continuum, if you will, where on the one side, you have diseases of high genetic clarity. This is where you have a genetic driver that we understand to be causing the disease. Those are typically rather rare, but they're also very easy to model in a cellular system. You can introduce the genetic driver into your cellular model, and our assay capabilities and machine learning allow us to understand the consequences of that driver in a cellular state, and thereby hopefully identify also things that revert that. On the other side are diseases that are much more diffuse and polygenic, still having a significant genetic basis, but driven by a dysregulation where multiple genetic causes come together. Those are harder to model correctly in cellular systems, but at the same time, they also tend to be much more prevalent, and therefore we can hope to use larger amounts of clinical data to tease apart the different causal factors. In the middle are diseases that have elements of both. These are diseases that on the one hand are polygenic and hence lend themselves to potentially larger amounts of clinical data, but at the same time have multiple strong genetic footholds where we have clear genetic drivers in small patient populations. The disease of ALS, which is one of the ones that we're very much front of mind for us, actively working on, is a poster child for that, because 90% of the ALS patients are sporadic, but there are 10% that are driven by strong familial mutations. And so ALS allows us to bring to bear both of the capabilities on this slide at once. And what's really exciting to us is as we genotype and phenotype increasingly larger patient cohorts, we are more and more understanding that most diseases are in fact somewhere in the middle. Even quote monogenic diseases such as Huntington's disease have a polygenic background that significantly affects disease outcomes, and even polygenic disease such as autism, schizophrenia, or obesity have small patient populations with strong genetic footholds. So for a lot of diseases, we will be able to deploy both types of capability. So as I mentioned, we have 3 therapeutic areas that we're working in: metabolism, neuroscience, and oncology. And in the rest of the talk, I will show 2 vignettes that help illustrate those capabilities. I will demonstrate our work in oncology focusing on clinical data and our work on neuroscience focusing on cellular data, just to give a sense for the spectrum. So here is the work in oncology, where, as I mentioned, the focus of our efforts is to try and uncover novel insights from large clinical cohorts. One of the cohorts that we have privileged access to and that we're very excited about is a cohort called Genomics England, which is a partnership that was announced just a little over a year ago, which is a partnership where we help them digitize and, importantly, also interrogate large amounts of histology data. So in this cohort, which is actually the evolution of the UK 100,000 Genomes Project, they have access to about 17,000 cancer patients for whom they have tumor and germline genetics, as well as longitudinal clinical outcomes through the National Health Service. The addition of histology images from these whole slides taken from these cancer patients provides an incredibly rich resource to understand much better the connection between the genetics and the downstream clinical outcomes. So while we were waiting for those data to become available, which I'm glad to say they now finally are, we started to work on a cohort that was much older and maybe not as high quality, as a way of developing capabilities to prepare for the GEL cohort. So that cohort is TCGA, the Cancer Genome Atlas, is a monumental effort that began in 2006. It has over 20,000 tumor normal samples spread across 33 cancer types. It is very broad, but also was built at a time that QC metrics were quite, and protocols were quite different across different academic centers. So it's a fairly disparate cohort with varying quality. I told the team as they started to work on this that it was certainly going to be, because of that, an interesting challenge problem in machine learning to address all of those artifacts and issues. But at the same time, they weren't likely to learn much new scientifically, because it's a cohort that's been mined quite extensively by thousands of researchers. It turns out I was right about the first, but wrong about the second, as I will now show you. So here we're learning the language of clinical data. So we start with high content data, in this case, histopathology, and other applications. As I said, we're working on MRI, blood biomarkers, and so on. And we're applying self-supervised deep learning, which is, again, a technology in the same spirit of what's currently used in the large language models, to create a low-dimensional embedding that captures the essence of what is in each of those images. And whereas each of those images is billions of pixels, the low-dimensional embedding is only a few hundreds. And that's important because you can then take that embedding and juxtapose it with other types of data, that other types of covariates that might exist only in smaller cohorts, but because the embedding is so much lower dimensional, as well as being quite semantically informative, you can, those associations between the embedding and those other covariates, whether clinical or genomic covariates, end up being well powered for discovery, and that turns out to be very important. So let me first demonstrate a couple of examples on clinical covariates. What you see here is basically what's called the Kaplan-Meier curve, which is a survival curve. So the x-axis is years, and the y-axis is the percentage of patients that are alive so and so many years from baseline. And this is, on the left, is a stratification of patients into high and low risk based on the standard covariates that are used as part of the standard of care. Age, sex, what's called IHC subtype, and tumor stage. This is in breast cancer. We have similar graphs in other cases as well. And so you can see a pretty reasonable stratification and what's called a hazard ratio of 2.19. On the right-hand side is what happens when you take the information from the histology embedding, trained in the self-supervised way, and adding that on top of those standard clinical covariates, and you get a much stronger stratification between high risk and low risk patients, with a better p-value and a stronger fit. So that's interesting. I think this graph is actually even more interesting because it shows the correlation between the risk score that you get from the embedding, the histology, and the risk score that you get from the clinical covariates. And what's noteworthy is that these are entirely uncorrelated. That is, the correlation is 0, which illustrates that there is considerable orthogonal value to be gained from the embedding beyond what a clinician currently knows to look for, demonstrating that there's a significant amount of additional value that can be gleaned. So that's really great and clearly has implications or potential implications in things like stratifying risk, predicting response to treatment, things like that, which you could do from the histology data. But what it doesn't directly give you is novel intervenable targets that could potentially help you cure additional patients. And so in order to get to that, what we ended up doing was going to a different type of data modality, which is molecular measurements that also exist in many cases, I would say in most cases, only at relatively small scale. So one of those assays that we got quite excited about is something called ATAC-seq, which I'm sure most of you have not heard of. It's actually a relatively new assay as these things go. It's about a decade old. And what it does is it measures chromatin accessibility, or in lay language, the openness or closeness of certain regions of the DNA, and hence whether they are likely active or not active. This is actually a very valuable assay because it allows you to see the activity of genes that are actually quite lowly expressed in terms of the amount of RNA and protein that they produce. Those tend to be drowned out in other types of assays, but by things that are much more highly expressed, but are often quite important from a clinical perspective. They also allow you to identify different kinds of changes that are not necessarily having to do with coding genes, and are often play a very significant role in cancer genesis. So ATAC-seq, as I said, is in many ways a wonderful assay, but the downside is that it's a very finicky one. It is very sensitive to how the samples are processed and will likely never be used clinically as part of the standard of care, and is available only in limited scale research data sets. Fortunately, one of those limited scale research data sets is this one. It was part of TCGA, so the same lab that invented ATAC-seq, the Howard Chang lab at Stanford, took 400 samples from TCGA and basically ran ATAC-seq on that. This is a paper by Courses et al. And so we basically had a data set of 400 patients spread peanut butter thin across 23 cancer types. And so the largest subcohort within that was, I think, breast cancer with about 30 samples, clearly not powered for any kind of meaningful analysis. However, the question that we asked ourselves was a radically ambitious one, which is to say, well, great, you have this 400 patients worth of ATAC-seq data. Can you use the histopathology, which is available for 11,000 patients across 31 cancer types, to impute the ATAC-seq profile, that is the openness and closeness of regions of the genome? And you might say, well, how on earth would you be able to read the openness and closeness of the chromatin from a microscope image? It turns out that certain genes actually manifest quite nicely in histological signatures. And all in all, we were able to actually predict about 5,000 peaks worth out of 500,000. So about 1% are predicted with a very significant R squared of greater than or equal to 0.5. We could actually do better than that, but this was sort of like we picked a fairly stringent threshold. And what you see on the right is the actual ATAC-seq on the left, the imputed or predicted ATAC-seq on the right. Needless to say, machine learning 101, these are on held out patients. So you're predicting on patients that you've not trained on. And you can see that there is a very strong similarity between the actual and the predicted, which is also quantitated on the bottom right in this correlation graph. So with that, you suddenly have imputed ATAC-seq over 11,000 patients, which is about 2 orders of magnitude more than you had from the original 400. So all of a sudden, you're now powered for some pretty impressive analysis. So what you see here are the top ATAC-seq peaks in terms of their association with overall survival. So and we simply took the peaks, we sorted them via their hazard ratio, which is the association with outcome, with survival, and this is uncherry-picked list sorted from top to bottom. And because most of those ATAC peaks are actually located in the close proximity of genes, you're able to associate them by and large unambiguously with gene names. And so what you see here is the genes that are underneath those peaks. And I can tell you, I'm sure many of you are not cancer biologists, but this is a list of what you see on the left is a list of greatest hits of breast cancer genes, alongside with 2 that are novel. They're novel in breast cancer, they're not novel in other solid tumors. And so that potentially suggests 2 intervenable nodes in breast cancer that didn't exist before. What's perhaps even more exciting is the graph on the right, which shows the same kind of analysis, but on triple-negative breast cancer. Triple-negative breast cancer, obviously, is a subset of breast cancer, so it's a smaller cohort. Breast cancer is about 1,100. Triple-neg is about 300. And what's notable is that even though it is a smaller cohort, both the hazard ratios and the p-values are considerably more significant, because in some ways this is a more coherent cohort, and so the signals tend to be much stronger despite the decreased power. Here again, we picked up 3 known cancer drivers in TNBC, as well as 5 novel ones. And I can tell you that of these 5 novel ones, again, there is support for them in other types of cancer, including breast cancer. They have not been previously identified for TNBC. TNBC is a disease that has considerable unmet need. Most of the patients today are treated via chemotherapy, which is really not a great drug. And so this potentially gives us novel intervenable targets in triple-negative breast cancer, which we're very, very excited about from the perspective of patient impact. What's shown here is another form of support for the TNBC targets. So also in the TCGA data set is whole tumor sequencing for those patients, for the tumors. And what you see here is a map of the somatic mutations, the regions in the DNA, or the genes rather, that are mutated in the cancer. When genes mutate, they can be amplified, which means increase in the number of copies. They can be deleted, a decrease, or missense, which is often hard to interpret. You can see here, just by looking at the patterns of red and blues, that these genes that we identified actually have considerable enrichment for amplification events, which aligns with what came out of the ATAC-seq, which is those regions are now open, which suggests that the genes are now active. And that really provides support for the causal role that activating these genes has in allowing the tumor to persist and proliferate, and hence have adverse patient outcome. So this is kind of an orthogonal form of support for the findings that came out of the ATAC-seq using a completely orthogonal data modality. So we're very excited about this, and we're now working to increase the conviction on those targets via additional forms of evidence and also additional cohorts, and then also using the wet lab capabilities that I'll show you in the next section to interrogate those targets and gain conviction around their therapeutic value. So summarizing the work that we've done in oncology, it's really a very differentiated strategy in that it uses a human-centric approach with primary human data at its core, avoiding the kind of reductionist model systems or non-translatable animal models that are typically used in the field. It takes the histology and uses unbiased machine learning to really capture the fundamental structure at multiple scales of the tumor microenvironment, including the interactions between one cell and another cell. And then using machine learning, we can impute additional covariates that help us provide additional layers of information, which allows us ultimately to put it all together and create a data-driven assessment of the impact of different things that we could do to the tumor, different genetic or genomic changes on clinical outcomes, which hopefully will give rise to novel targets and novel biomarkers. So moving on from this to our work in neuroscience, which is, as I said, uses a very complementary and distinct set of tools. Here we're using cellular systems that we build in-house in our fairly sort of cutting-edge wet lab capability. I'll show you a video in just a moment. So here we take cellular models. In this case, they're actually neurons that are differentiated from induced pluripotent stem cells. Induced pluripotent stem cells are cells that are taken from individuals like you and me. They're reverted back to their stem cell status using reprogramming, a technology for which Yamanaka won the Nobel Prize a few years ago. And then they are differentiated back into neurons, and we can then introduce known disease-causing mutations into those neurons in order to see the difference between healthy and sick, where sick is defined genetically. We take those different populations, healthy and sick, we measure them using a range of different multimodal assays. I'll talk about that in a little bit. And then on top of that self-supervised learned representation of cell state, we overlay a disease axis that goes from the sick to the healthy, which allows us to potentially identify things, interventions that revert sick cells back to healthy. So let me talk a little bit about some of the more innovative assays that we've developed as part of this work. This is a proprietary microscope that uses what's called quantitative phase imaging. Quantitative phase is a technology that measures not only the amplitude of light, but also the phase, the angle, if you will, of the light. It turns out that light, when it shines through a translucent object like cells, depending on what it hits, it will refract at a different angle. And so it will refract in a different way if it hits, say, a lipid, a fat, versus something else. And so what we do here, you can see on the top left, this is a rotating panel of LED lights that allow us to measure the angle of refraction when the light shines through the cells at different angles. We use one layer of machine learning to put all of that together to create a single integrated high contrast, high resolution image of the cells, which is what you see at the bottom. Now, on top of that, we trained an additional machine learning model that was trained by taking the output of that first machine learning model and then we fix and stain the cells. We measure the phase microscopy using the existing microscope. Then we use laser to stimulate the cells so that you can see a fluorescent marker that stains or tags particular cellular compartments like nucleus or chromatin or synapses. And so you can think of the top as the input to a machine learning model, the bottom is the target output. We train a machine learning model to impute the stains from the QPI output, which basically allows us to hallucinate, kind of like a deep fake, stains on top of the cells. Now, this has a couple of different repercussions. First of all, is that you don't need to stain the cells. Fixing and staining cells, it causes considerable artifacts to the cells. The cells change as a consequence of that, and we don't have to do that. But more importantly, you can do it without actually stopping the experiment. You can do it on top of the live cells. And so you basically get the ability to measure all of these different cellular compartments as part of a longitudinal experiment, which gives you much more robust and richer phenotypes, because you can see things, as I'll show on the next slide, of like things like lipid droplets moving into and out of the cells, and where they move in the cells and how the droplets form and all sorts of really cool things like that. So this is an example of some of the lipid phenotypes. What you see at the top is the reconstructed stains. What you see at the bottom is what happens when you fix and stain the cells. And again, it's basically visually indistinguishable. And now, as I said, you can start to do all sorts of really interesting measurements on top of these cells. So this is some of the work that we did in metabolism. It's not the focus of the section, but it really helps highlight that you can introduce different concentrations of free fatty acids into the medium that the cells are in. Think of it as the McDonald's diet for cells. And then as you introduce more of that, the cells develop more and more fattiness. You can see the little shiny droplets inside the reconstructed QPI. And it turns out that the self-supervised learning just picks out that pattern beautifully without actually even being told what medium the cells were exposed to. And you can basically get this beautiful dose response of the number of McDonald's diets that the cells got, and it picks out that gradient by itself. So this is one of the ways that we have to discover a cellular phenotype that distinguishes disease from healthy. And what I showed you a moment ago was effectively a disease gradient. But now you need to be able to screen for reversions in that disease phenotype. And how do you do that in a scalable way? So what we did in parallel over the last few years is to build out a very complicated assay technology that allows you to do pooled optical screening using in a genetic way. So what does that mean? You take a population of cells, let's say neurons or hepatocytes or whatever, and you expose them to a library of CRISPR guides that basically are a technology that allows us to edit the cells. Each guide knocks out a particular gene. That library is introduced as a pool, but in a way that with a quantity that the average cell gets about one guide. And so you end up at the end of it with a genetically diverse population of cells where each cell got one guide that knocked out one gene. And so you kind of have like a population of little knockout cells. you can now use a microscope to measure a diverse set of phenotypes on those cells, ranging from core cell morphology, using some of the staining methods that I talked about before. You can measure the localization of specific proteins within the cells, and you can do this live cell imaging that I mentioned earlier. And if you look carefully at this panel on the bottom right here, you can actually see little dots that are moving from one neuron to the other. These are mitochondria moving across axons, which is like really cool that you could see that. Having phenotyped all of those different cells at the single cell level, we then fix the cells, and then we do what's called in situ sequencing, because each of those CRISPR guides is tagged with a unique DNA barcode. And so we basically built a little sequencer within the microscope that sequences the barcodes one base pair at a time, so that we can tell which gene got which guide and hence what genetic intervention it got. And that allows us to build a really rich matrix of individual cells, what gene got knocked out, and then a very rich phenotypic readout that uses all of these different forms of imaging. And so that is a very effective way for measuring genotype-phenotype relationships. Now, building that was only the first step. We also needed to productionize it. And so a very extensive amount of work went into everything from microscopy, chemistry, cell biology, automation, lots of data pipelines, and an incredible amount of machine learning, because it turns out that without machine learning, you can't even segment the cells and appropriately associate the barcode to the cell. And so machine learning is kind of used here also as our Swiss Army knife, if you will, not only as the ultimate readout technology for the data. And so this is, so this is basically just an illustration of what this is what the input looks like, and then the in situ sequencing, which you can see one base pair at a time being read out, and the machine learning kind of puts it all together with a sort of assignment of a barcode to each cell. And I think this is what I already said, which is that the machine learning really plays a critical role in processing these pooled optical readouts on multiple different dimensions, including the barcode sequencing, correcting for illumination and batch effects, identifying cells and nuclei, and so on and so forth. Okay. So one therapeutic indication for which we've used this is a devastating disease called TSC, or tuberous sclerosis complex. The TSC Alliance is a patient advocacy group. They visited us actually quite recently at In Sitro, and we heard the heart-wrenching stories about the patients of this disease. So let me tell you about this disease. It is a roughly monogenic disease. It's caused by a loss of function in one of two genes, TSC1 or TSC2, that form a complex. When one of those two genes is lost, it leads to an activation of a core nutrient sensing pathway called mTOR. That ultimately gives rise to a set of symptoms that roughly correspond to the creation of these, quote, benign tumors that spread all over the body. These are benign, not in the sense that they're nice, but in the sense that they're not metastatic. And when they occur, they can have significant morbidities, specifically when they occur in the CNS, the central nervous system. The most common symptom is refractory epilepsy and neurological impairment and intellectual disability, which is largely correlated with the seizures. And we're talking about something like 40 seizures a day for some patients. So it's really a horrible disease. The most commonly applied therapeutic, though, is something called everolimus, which is an analog of rapamycin, which inhibits the mTOR pathway. It is effective in modulating the seizures in about 50% of patients. And so you can see that there's considerable unmet need in terms of alternative therapeutics. Moreover, this disease is also in some ways a real proof point or a launch pad for other diseases that of a similar nature, such as other genetic epilepsies and other early neurodevelopmental disorders. So we constructed these disease models that I mentioned earlier, using the TSC knockout as the disease, the healthy cells as the wild type. and then basically constructed a set of disease models that use the various phenotypic modalities that we have in-house. This is a RNA sequencing disease model that uses transcriptional signatures, RNA transcript signatures. And in order to gain conviction around the fact that this is truly modeling disease, I mean, separating the sick cells from the healthy cells is a nice start, but it's not necessarily something that gives us complete conviction. So here we took this rapamycin treatment that we know to be an effective clinical intervention, and we basically saw that same intervention returns the cells also from the predicted disease state to the predicted healthy state. So that creates an alignment, if you will, between cell state and human clinical state. On the right-hand side is a different form of validation, which is we were fortunate to be able to get RNA sequencing data, in this case in bulk, from the brains of patients, the TSC patients on the right and control healthy patients on the left. And you can see that the model that was trained entirely on the TSC cellular model perfectly separates the patients from the wild type. And so again, providing us conviction around the disease model. We also used our imaging technology in order to create a disease model. So this is some of that live cell imaging that I showed earlier. And again, you can see on the right a disease axis, reversion across the disease axis by different doses of rapamycin. And so where the top is the TSC, the bottom is the wild type, and different doses of rapamycin get you closer and closer to the wild type cells. So again, giving you conviction around the disease axis. So we took all of these different disease models and we basically screened them using our screening technology that I mentioned earlier. And this is using a machine learning technology that tries to separate movement across the disease axis from orthogonal movement, something called OPLS, and identify genes that are specifically moving you along the disease axis from the sick to the healthy state. And all in all, that gave us about 60-some hits at this point. At this point of the work, it was actually 40. We've gotten a few more since then. So 40 of those screening hits went subsequent validation to try and pick the ones that we want to actually pursue as potential drug targets for this devastating disease. So one form of validation that we have a lot of conviction around is support in human genetics. There's considerable evidence at this point that genes that have support in human genetics are about twice as likely to be successful as ones that do not. And so what you see on the left here is the 40 hits mapped across whether they do or do not have support in human genetics for the different traits that are related to TSC. And so you can see that about 60% of our hits have support in human genetics. And what's notable is that the majority of the support comes from epilepsy and other neurological diseases, such as neurological impairment. This is relevant because it turns out that we did the screening in neurons. There were other previous screens as well as in our own hands that were done in other kinds of cells, specifically various random kinds of cancer cells. The set of hits that you get are quite different, and they do not similarly overlap with phenotypic traits that are neurological in order. So that in nature. So what that really supports is the hypothesis that it really matters what cell type you screen in order to uncover meaningful modifiers. The other form of validation that we're doing is one that uses a functional readout, something called MEA, or multi-electrode array. This is a very complex experiment. You would not use it for primary screening, but you can use it for validation. This is cells that are grown for 49 days, and as the neurons mature, they start to connect to each other and they generate network activity. And for the TSC patients, that network activity is disrupted. So TSC knockout cells have a different form of network activity than wild-type neurons do, in a way that very closely mirrors the kind of dysfunction that you get in the EEG of TSC patients that are undergoing a seizure. And so you can see that this is a phenotype that is genetically associated with the disease. And furthermore, that phenotype is also reverted by rapamycin, which again gives us conviction about its translatability. And so we took a subset of the hits that came out of our screens. We started out with 15 that had chemical compounds that one could use for the experiments, because that's easier than trying to intervene genetically. And we tested 15 highly ranked hits in the MEA assay with tool compounds. And we were honestly blown away, because I expected maybe a couple of those to revert an MEA phenotype, but it turns out that 7 of the hits, including 6 that are very novel, had not previously been associated with the mTOR pathway, reverted this hyperexcitability phenotype. So if you look at what this graph shows, the red is the firing rate, which is one of those hyperexcitability phenotypes that you get. This is the TSC knockout. The green bar is the wild type, so very different. The blue is the reversion you get from rapamycin, and the purple bars correspond to the dose responsive reversion that we get for our tool compound. You can see the more drug you give, the more reversion you get, to the point that you get a reversion that's commensurate with that of rapamycin. And so we're very excited about this, both because it gave us a set of meaningful novel targets for this horrible disease, and also provides what is a very impressive validation of the platform, because this is a completely orthogonal assay. The original experiment was done, the ones that identified the screening hits was done on 2-week-old neurons using an imaging assay. And this is 49-day neurons on a live cell morphological readout. I was expecting maybe a 10% hit rate. We got close to 50%, and we're very excited about that. So this technology is now being used in a whole slew of other diseases that we're interrogating. One of the ones that we spent the most work on is ALS. I mentioned that earlier. This is a disease where about 10% of the cases are familial, caused by known genetic mutations. The remainder are sporadic, and it's not clear how they emerge. This is actually a fairly, reasonably common in the sense of, these diseases in terms of neurological diseases, about 6,000 new diagnoses each year. And it basically, it's a progressive disease that eventually results in paralysis of nearly all of the skeletal muscles, and the death typically occurs between 3 to 5 years of onset, mostly because of respiratory failure. So there's a few drugs out there, not many, and they only very slightly slow the progression of the disease. So in order to interrogate ALS, which is a much more complex disease than TSC, we've created the largest known library of isogenic iPSC lines that allow us to model the spectrum of genetics that give rise to ALS, both familial as well as sporadic strains. So for the familial, we engineered in a wild type background the disease relative to the wild type, which gives us a very clean case control. For the sporadics, of course, we onboarded patient lines. We also developed state-of-the-art capability to robustly take these iPSC lines and generate motor neurons in a completely automated way, which is absolutely essential both from a scale as well as a quality perspective. And then all of the phenotyping work that we did on TSC, but with additional ALS-specific modalities, is used to interrogate those cells. And the idea here is that we actually would be able to take the different genetic backgrounds, which ultimately they all give rise to motor neuron death, but probably via different trajectories because of the different pathways in which they occur, and perhaps use the high content phenotyping that we have to identify those coherent patient subsets and relevant reversions in each of them. And so this work is actually going on right now. We're starting to do screens, and we're very excited about the prospects for this work. So here what we're doing is we're taking these human-derived biologically relevant cellular systems at unprecedented scale. We are generating multimodal phenotypes that allow us to unravel the heterogeneity of those diseases across diverse genetic backgrounds. We're using machine learning to identify healthy and the different kinds of sick cell states, align that with human clinical outcomes, and then use these pooled genetic screens to identify effective intervention points, which will be the starting point for a drug discovery program. So all in all, what I've described to you is a platform that has been in the making for almost 5 years. It's a multi-part, fairly complex platform that has biology, it has chemistry that I didn't get a chance to talk about, generating in vitro data at scale, juxtaposed with the acquisition of clinical data across modalities. All of that feeds into a coherent, unified data platform on top of which sit a set of machine learning algorithms that allow us to analyze the data and generate insights that provide us opportunities in terms of novel targets, patients, or therapeutics. And what's important is that this is a powerful platform already, but it will continue to get more and more powerful over time as we develop additional ways of generating data, additional data partners. More data will flow into our data stores, which will enable even more advanced machine learning to be applied, because we know that the more data you have, the more elaborate and complex the machine learning methods that you can use, and that will give us more high confidence biological insights as well as targets, which will give rise to a growing pipeline of high quality therapeutic interventions. So all in all, this helps enable our long-term aspiration, which I think, like many in the biopharma world, is to discover and develop transformative medicines for patients. But for us, this is only one part of a three-part goal, which is we also want to do this in a way that de-risks and accelerates the standard R&D process via these predictive models that are based on machine learning and data at scale. And importantly, to do it in a way that is engineered so that it's not just something you do once or twice, but it really provides you with an engine or a flywheel that allows us to do this faster and better over time, especially given that machine learning just keeps getting better the more data you feed it. And so it naturally gives rise to this flywheel effect. So with that, let me just maybe end on a slightly philosophical note, which is, there's many really exciting opportunities today to apply machine learning in a whole slew of different areas. I mean, it's an incredibly exciting time for machine learning and AI, but let me maybe try and convince you that applying machine learning to biology is a particularly exciting place. If I think back on the history of science, there are epochs, particular periods of time when one discipline really made a tremendous amount of progress in a short amount of time. It was chemistry in the 1800s with the periodic table, physics in the 1900s with the relationship between matter and energy and space and time, computing, of course, in the 1950s. Then in the 1990s, we actually had two areas that started to evolve: data, what gave rise to current day machine learning, and quantitative biology. I think one of the critical disciplines in the 2020s will be what I call digital biology, which is the synthesis of these two disciplines that have been evolving largely independently, which is machine learning and biology, and will allow us to read the biology in whole new ways, interrogate what we see using machine learning, and then write biology using also an increasing range of tools. And that has implications not only in human health, but also in the environment, energy, biomaterials, agriculture, and many other areas that will really make a difference to life on this planet. So with that, I will end here and thank the incredible group of in situ sites that did all of this work. Thank you very much.